Folate and autophagy operate in the same cellular neighborhoods without often being discussed together in nutritional science communication. Autophagy is responsible for removing the damaged, dysfunctional cellular components that would otherwise accumulate and drive cells toward pathological transformation. Folate, through its role in one-carbon metabolism and DNA synthesis, provides the nucleotide building blocks that newly generated cells require after autophagy has cleared the way. The two processes are not competing, they are sequentially collaborative partners in cellular renewal, and the tissues where this collaboration matters most are precisely those with the highest cell turnover rates: intestinal epithelium, skin, and bone marrow. Intestinal epithelial cells complete their full turnover cycle in approximately 3 to 5 days. This extraordinary rate of cellular replacement, among the fastest in the human body, requires both continuous autophagy to remove damaged cells from the cryptovillous axis and continuous folate availability to synthesize the DNA needed for new cell production. Deficiency in either process produces the same tissue-level outcome: compromised epithelial integrity, increased intestinal permeability, and impaired barrier function against pathogens and dietary antigens. This is one reason that folate deficiency and gut permeability share epidemiological associations. They are connected through the coordinated biology of tissue renewal. The MTHFR gene variant deserves specific attention because it affects a significant proportion of the population. Estimates suggest roughly 40% of individuals carry at least one allele of the C677T polymorphism, and it meaningfully impairs the conversion of dietary folate and supplemental folic acid to the active methyl tetrahydrofolate form that participates in the methylation cycle. People with this variant have reduced methylation capacity, which affects not only DNA synthesis, but also the methyl group supply needed for epigenetic regulation of autophagy genes and the homocysteine recycling that B12 and folate share responsibility for. For these individuals, dietary folate from whole foods, which is predominantly in bioavailable reduced forms, is preferable to synthetic folic acid and 5-methyl tetrahydrofolate, 5-MTHFR. Supplements bypass the conversion step entirely. Lentils are among the most concentrated dietary folate sources at 358 micrograms per cooked cup, followed by spinach at 260 micrograms per cup cooked and asparagus at 262 micrograms. Liver provides 215 micrograms per 3-ounce serving. The recommended daily intake of 400 micrograms increases to 600 to 800 micrograms during pregnancy, when folate demand for fetal neural tube development reaches its most critical level. The risk of neural tube defects from folate insufficiency in early pregnancy is one of the most clearly established nutrition-health relationships in clinical medicine, and the first 4 weeks of pregnancy, when most women don't yet know they are pregnant, are when folate protection is most urgently needed. The critical warning that practitioners must communicate clearly is that high folic acid supplementation can mask vitamin B12 deficiency. Folic acid corrects the macrocytic anemia associated with B12 deficiency without correcting the neurological damage, creating a situation where neurological deterioration proceeds without the hematological signal that would otherwise prompt investigation. Anyone taking high-dose folic acid supplements should ensure B12 status is simultaneously verified.